21:15. So before watching this lecture, you should have completed all of the macromolecules, especially proteins. So what this lecture is going to be about are the different levels of protein structure. This is found in Mater, Chapter 3, Section 4. So just a little bit more detail on that. So skills to master, a lot of times I'm asked, okay, here's the lectures, what's going to be on the exam? These 3 things. If you can do these 3 things that from this after watching this lecture, then you're going to be fine on the exam. So if I give you a pic, if you can identify the level of protein structure, you're on the right track. If you have to list which level of protein structure is not found in all, so we're going to go over that. And you should be able to predict how changing primary structure could affect other levels of protein structure. So those are our 3 tasks that we should be able to do to be ready for the exam. Okay, so there's 4 levels of protein structure. There's primary structure, which is abbreviated as 1 with a degree. And then there's secondary structure, 2 with a degree sign. This is secondary. You're going to love these names. 3 with a degree is tertiary. And 4 with a degree is quaternary. So those are our 4 levels of protein structure. So starting with number 1, primary. And so it's so easy, right? 1, 2, 3, 4. So 1, primary. What this means is that this is the sequence of amino acids in an actual protein structure. Sequence of amino acid side chains, right? Because amino acids are made of the backbone and the side chain. The backbone's the same in every single one. The side chains are different. So that's what we mean when we're saying the sequence of a protein. So this is basically the sequence of a protein. So like your DNA sequence makes you you, the sequence of the protein is what makes the protein the protein. And this is how it's able to actually function. So if you were to write this, because of the sequence of the side chains, and remember from previous lectures, there's 20 different ones, 20 different amino acids. And so these can be abbreviated a few different ways. There's a 3-letter abbreviation. And so that's in the format X, capital letter, little letter, little letter. So everyone's favorite, glycine, this amino acid, it looks like this. H2N, here's the backbone coming at you, and that could be connected in a protein or not. Its side chain is the letter H. And that could be connected to the next one. C double bond O. And here we'll just do 3. C double bond O. Okay. So we're going to have a tripeptide here. H. There. Everything's correct. So glycine. So this has an H side chain, GLY. This is the 3-letter abbreviation. There's also a 1-letter abbreviation for that, and that's the letter G. And then you could draw out the side chains like we've shown here, or you could draw it as circles. So this one's 3 amino acids long. We could draw 3 little circles. So over here we've got this amino group, so this tells us something important. The amino group's hanging off on this side, and this carboxylate group is hanging off on this side, and everything else is the peptide bonds. So then you could put the symbols in here. So generally it's the 3-letter symbols in here. So let's do glycine, let's do serine, let's do how about threonine? THR. Okay, so this protein could be abbreviated GLY, SER, THR, just as text. It could be shown to you as circles with 3 letters in them, or 1 letters in them. Or it could be written as, let me scroll down for a second, or this could be abbreviated as GST. T. So I could show you this, and that's primary structure. I could show you this, and it's primary structure. I could show you this, it's still primary structure. Or I could spell it out like this. S-E-R-T-H-R. Still primary structure. This is just telling us what order the side chains come in. Okay. So now let's talk about secondary structure. Scroll down a bit. So secondary structure is based on primary structure. So here we go. Secondary. So the sequence of the amino acids, the primary structure, tells us what the secondary structure is going to look like. And there's actually two motifs that you can look for that are secondary structure. And it's called, one is called the alpha helix, and the second one is called the beta strand. So these are the first two letters of the Greek alphabet, alpha and beta. So the alpha helix looks like a ribbon, looks like a curlicue, right? And the beta strand is most of the time depicted as an arrow. Okay, so when you have a protein sequence, whatever that protein sequence is, I'm just writing X's. Here's your primary structure. Primary structure. Depending on what these side chains are, you're going to have close interactions, and that's what this is. These are close interactions of the side chains, or these are far away interactions. And then there's technically a third one called loops, and that's no interaction. But that doesn't really look like anything on the paper, it just looks like a line. So that's what's going on there. So then maybe these would form a helix, and maybe these would form a beta strand. So it's just like a list of how these amino acids would be arranged based on their interaction. So this is secondary structure, what that would look like. So on the exam, I'll show you either something that looks like this, or something that looks like this, or something that looks like that. And that's what's going to be the secondary structure. So you're looking for curlicues, you're looking for arrows. Two parts of secondary structure. Okay. So now, let's talk about tertiary structure. So tertiary structure is based on the secondary structure, and because it's based on secondary structure, it's also based on primary structure. So this is how these side chains interact with each other and how their backbones interact, and this is how the alpha helices and the beta strands interact with other parts of the protein. So the third level is the 3D shape. You like that? Third level, 3D shape. Second level, two motifs. First level, first thing you should think of, the sequence. How nice is this? So this is the 3D shape of a protein. And the different structures actually give it different functions. So there's two types of structures that we're going to talk about. Globular structures. And this is found in a special type of protein called enzymes. So these really look like a big glob. So they're kind of like circular, and they have a special region right here that's called the active site. And this is where some sugar or some lipid or some chemical is going to bind here, and that these are going to perform chemistry. So enzymes are a special type of protein that perform chemistry. So chemistry is done by enzymes. And there's another type of structure called fibrous. Fibrous structure. So these look like big fibers, and they can be sometimes braided together. Oh, here, we'll talk about that in a minute. So these look like big long fibers. And so there's a whole bunch of maybe alpha helices interacting here. And what these are for, because these don't have an active site. You see, here's an active site, no active site here. What these are for is these are for structure. So this is what your cytoskeleton's made of. Fibrous, like long, these are rope-like proteins. So these are used for structure, like the cytoskeleton, like your hair. So when you're thinking fibrous, think of your hair. Okay, and then the final level is quaternary. And this, as you would predict, is based on tertiary, which is based on secondary, which is based on primary. And so what this is interactions of more than one, so at least two, 3D protein. So we see this in this protein called hemoglobin, which is globular. So these are our globular proteins. Hemoglobin is found in your blood, and it does its job of carrying oxygen, which is O2. So each subunit will bind oxygen. So this is what a globular quaternary structure would look like. Hemoglobin. Hemoglobin. Okay, and so each one of these circles here represents a protein chain. So we could call it chain A and chain B. So on the exam, of course, I'm going to have to label these as different proteins. And I'll show you actual crystal structures of actual proteins during class. This is just my rough sketches. Okay, so if we have globular quaternary structure, we should also have fibrous quaternary structure. And so that's what I tried to draw before there. So as you're adding more and more, it's going to kind of look like a braid. So here's chain A, and of course they're always going to be labeled, and here's chain B, and maybe it's going behind and wrapping around. And you can imagine that lots and lots of fibrous proteins actually have quaternary structure. But as you can also imagine, some globular proteins, some enzymes, maybe they don't need other proteins to work. So this quaternary structure is something that not all enzymes have. Not all proteins have this. So this is the level that not all proteins have. Okay. So the final skill that you should be able to master is how would an effect of primary structure, how would changing the primary structure affect quaternary structure, affect tertiary structure, affect secondary structure. And so we're going to work on this more in class, changes to primary. And I'm going to say it depends on the change. Okay, because as you're changing the side chains, maybe you have a side chain that looks like this, and you change the side chain to look like this. It's basically the same thing. So this would be hydrophilic, and this would also be hydrophilic because it's got a polar part. So this type of change, hydrophilic to hydrophilic, would actually, you wouldn't expect that to change the secondary structure, or the tertiary structure, or the quaternary structure if it has one. However, if you take something hydrophilic and you change it to something hydrophobic, so now this doesn't have that OH group anymore, this would have a big change on the secondary structure, on the tertiary structure, on the quaternary structure if it has one. So these, if you're keeping the side chains the same, like hydrophilic to hydrophilic or hydrophobic to hydrophobic, it's not going to affect any other level. It's just going to affect primary. Because if you're changing primary, it is tautological that primary will change. So just keep that in mind. So these types, hydrophilic to hydrophilic, hydrophobic to hydrophobic, not going to change anything else, just changes primary. These hydrophilic to hydrophobic, or vice versa, hydrophobic to hydrophilic, this would change all levels. If it's a big change, it changes everything. Every level. Because changing the interactions is going to change the side chains, of course, which is going to change the secondary structure, which is going to change the tertiary structure, which is going to change the quaternary structure. And we're going to go over some more examples of this during class.